Hi, and welcome to Mr. Fisher's Biology Podcasts. This podcast deals with mutations. So, within DNA, during protein synthesis or DNA replication, nucleotides can be lost, rearranged in error, and that can result in a protein that doesn't function. And this is the basis to a mutation. So, mutation is a change in an organism resulting from a chemical change in their genes. So in other words, when the DNA changes, then we have a mutation. Now, we can identify three main types of errors or mutations that can occur. The first of those is called a substitution, or also called a point mutation. And the second two are deletions and insertions, and they're both grouped into this frame shift mutation group. So a point mutation occurs when one base from an original DNA strand is switched with another. So this can have multiple effects. The first effect is a non-functional protein. So for example, it could result in a stop codon being produced, ending the protein. Sometimes substitutions are silent or they have no effect. So for example, if we were to substitute a G in CCG to an adenine in CCA, looking at our DNA code wheel, we would find that they both code for glycine, so in other words, there's no real effect. And so that's a silent mutation. So again, looking at an example, we can have no effect on an amino acid sequence. So in this case, we've taken this C and we substituted for a U. The end result, though, is still the same amino acid. So this is known as a silent mutation. On the other hand, we can have what's called a missense mutation. And a missense mutation is typically when that mutation occurs within the first or second nucleotide on a codon, we can drastically change the amino acid sequence. And in this case, notice we haven't really changed it that much. We've replaced one amino acid with the other, but this can result in a non-functional protein. And that's an important point to note. Now, frame shifts, which include our insertions and deletions, these are more serious. What happens in these is that we either get the addition in an insertion or the reduction in a deletion of one or more base pairs in a gene. So this results in a shift in the reading of the bases, meaning that in often cases we get what's called a nonsense read. What happens is all the triplets downstream from the error code for the wrong amino acid, and this will occur unless the deletion happens or the insertion happens to include multiples of 3. So to better understand, let's look at an analogy. If we were to consider an insertion or a deletion in terms of a sentence in English, so this is the original sentence. Now if I were to delete the G in this sentence and then shift all the letters so that they're still in groups of 3, notice that the stays the same because it's upstream, but everything else has dramatically changed and becomes nonsense. Same is true in a DNA strand. So to look at a couple examples, by the way, when we talk about wild types, what we mean is normal in nature. So that's what we would find in nature. And so in this case, we're deleting a uracil out of this amino acid here. And that causes a shift in the reading, which not only changes the amino acid sequence, now we have a leucine and an alanine, but notice where we had a stop, we no longer have a stop. So our ribosome is going to keep producing until it reaches another stop. And this is going to be very bad. Okay. Now, in some cases, we can have an insertion or a deletion that could cause immediate nonsense. So in this case, our original DNA strand produced this mRNA, which produced this amino acid sequence. Now notice that after the methionine, there is an insertion of a U. So a U gets inserted right here. what happens is we've now created a stop codon where we should have 3 other amino acids. The ribosome will stop producing amino acids, and we will have no protein formed. Now, the effects of mutation. Mutations can have no real change. They can produce some minimal or drastic alterations. So some notable diseases like sickle cell anemia is caused by a substitution of one base. In some very rare situations, mutations are beneficial to an organism. Very frequently, though, mutations are lethal. So mutations are bad, but mutations are essential when we start talking about evolutionary change, because the only way we get new alleles, the formation of new alleles, is through the mutations in DNA causing beneficial changes to an organism and causing that organism to better adapt to their environment. Okay, so that in that case mutations can be good, but this is very rare. Just to review, we can have some different types of mutations again. So we can have our insertions or deletions. So that's a deletion. We have the immediate nonsense. We can have, in this case, we can have a frame shift that causes very little in terms of effect. It may drastically change the protein, but it doesn't cause a nonsense read. We've only changed an amino acid. We can have no effect on the amino acid sequence. These are our silent. We can have missense, in where we maybe substitute in and change one amino acid. This is that sickle cell anemia. This is what happens in that. And we can have a nonsense read, so we can substitute out an amino acid and cause a stop codon. So that concludes our podcast on mutations, and I hope that was helpful.